Hi, everyone. Let's get right into unit number 2, which is metabolic processes. The main topic in this unit is cellular respiration, which is exemplified by the chemical reaction on the top of this slide. We know it is cellular respiration because it starts with glucose and results in the production of energy. Just to tie it all together, we have to remember that the glucose that is required to start cellular respiration actually comes from the product of photosynthesis, which is shown in the chemical equation at the bottom here. Organisms may acquire this glucose directly or indirectly through primary producers that have the ability to photosynthesize. So what is cellular respiration? Cellular respiration, the process as a whole, okay, there's 4 different stages to cellular respiration, but the process as a whole requires oxygen. You can see that little oxygen molecule O2. Remember, it's diatomic. It readily binds to itself, so often it's in the form O2 in the atmosphere. And it uses chemical energy from glucose to make adenosine triphosphate. And that is the chemical energy that the cells can utilize to help metabolize all the processes in the cells and in the body. So we end up with we start with one glucose molecule, and it's showing down below here in that little simple equation, not even an equation. We start with the glucose molecule in cellular respiration, and at the very end of cellular respiration, we generate 36 ATP molecules. Okay, that's a lot. However, our body requires a lot to do all the work that it does. So we do generate a lot of ATP from that one simple glucose molecule. And chemical energy is now stored in ATP for use throughout the body. Here is a diagram of cellular respiration. A bit more detail. We're going to go into a lot more detail very soon. Okay, as you can see that this happens specifically in eukaryotic cells because it requires the membrane-bound organelle that is the mitochondria. However, the first stage is glycolysis, and this happens within the cytoplasm. So very primitive forms of life were able to generate small amounts of ATP by completing just the first stage of cellular respiration, glycolysis, because it doesn't require any specialized organelles there. But you can see, so number 1 in that pink square is glycolysis. It transforms glucose into a compound called pyruvate, and it generates some ATP. You can see there. Stage number 2 is pyruvate oxidation. That's in the blue box there. We can see that we have entered the mitochondria for stage 2, 3, and 4. Okay, this is where we take the pyruvate from the end of glycolysis and it transforms into acetyl-CoA. And then in stage 3, we're still in the inner matrix of the mitochondria where we have a Krebs cycle. It's cyclical. And this is where we get more ATP from. And then the 4th stage actually happens along the inner fold of the mitochondria within the cristae. And that's electron transport and chemiosmosis. They go hand in hand, and that generates more ATP for the end result of 36, as mentioned before. Okay, so this is really simplified, but it does show the reactants and the products within each broken down process and where they are located. So this is kind of handy if you get stuck with all of the details as we move forward. You can always come back to this to ground yourself, to think of the big picture once again. Here's another great diagram. This shows the 4 main stages and has a little bit more information attached, which will help summarize things as we go. Okay, so I'll start with this. I'd like you to, at the end of each stage, kind of come back to this to remind yourself, okay, think back to the big picture. How do these all tie in together? Because we could get very lost in the detail. So stage 1, as I mentioned, glycolysis. This is the only stage that is anaerobic. Again, this is when really primitive life forms were able to produce small amounts of ATP in the cytoplasm. So it actually doesn't require oxygen, although the rest of cellular respiration does, which is why we do say it is anaerobic as a whole. So lipolysis, which we're going to get into today, has 10 steps in the process for glucose to be able to be transformed into pyruvate. Then stage 2 is a transition reaction. Different sources call it different things. Sometimes they'll call it oxidative decarboxylation. I think our textbook uses pyruvate oxidation. But this is a one-step process within the mitochondria, and pyruvate is converted to a molecule called acetyl-CoA, which releases CO2, which it shows in that diagram right there. Stage 3 is the Krebs cycle. So this is cyclical. It keeps happening. This is an 8-step process within the mitochondria. It takes that acetyl-CoA from stage 2 and it oxidizes it to carbon dioxide. And this is where quite a bit of ATP is released, as well as coenzymes that are required for the 4th and final step, which is the electron transport chain and chemiosmosis. There's multiple steps involved. This happens along the inner fold of the mitochondria, so in that inner membrane there. And it uses those coenzymes NADH and FADH2 to create an electrochemical proton gradient, which powers ATP synthesis. Okay, so it's going to generate a lot more ATP in the end to all total 36 ATP at the end of cellular respiration. Let's just recap a little bit of background information here. So there are energy transfer mechanisms that can be used by this cell. How do we transfer energy into the cell? And there are 2 methods, which we'll be talking about a lot in these processes. So the first one is substrate level phosphorylation. So this is a method of forming ATP directly in an enzyme catalyzed reaction. So this is where the adenine triphosphate with 3 phosphates hanging off of it phosphorylates or detaches the phosphate, which breaking that bond provides energy, and it catalyzes a reaction upon whatever substrate it is attaching the phosphate to. Okay, so you can see at the bottom there, there is substrate, that bit blue, somewhat triangular substrate that is found in the active site. And you can see that the adenosine, what used to be triphosphate, you can see 3 Ps there sitting in the active site, but you can see that there has been a bond that broke off of the last phosphate group, and it has formed on that substrate. It is now attached. However, the ATP is now labeled as ADP, diphosphate. Okay, and I really want you, whenever you see diagrams, which we'll be looking at a lot again, when you're looking at compound names, if it says ADP, that means that it's adenosine diphosphate, and there are only 2 phosphate groups. To be able to harness and release energy, you need ATP to come in and then release the phosphate group. Okay. There's also something called oxidative phosphorylation. And oxidative phosphorylation is a method of making ATP indirectly through a series of enzyme-catalyzed redox reactions involving oxygen as the final electron acceptor. So being able to gain and lose electrons. Oftentimes, textbooks might say a hydrogen atom, which has 1 electron. Okay, so sometimes they'll talk about hydrogen atoms. This is a gain or loss of 1 electron, essentially. right? Because it only has one electron on in its only orbit. So sometimes they'll use hydrogen interchangeably when they talk about oxidative phosphorylation. So let's talk about NAD. So long form is nicotinamide adenine dinucleotide. Now, oxidative phosphorylation begins with the NAD plus molecule picking up electrons and passing them to a number of sequential redox reactions, with oxygen being the final electron acceptor, in which case, when it's finally accepting these electrons that keep bouncing back and forth in the redox reactions, which are often coupled, in the end, there will be an oxygen that finally accepts it, and that's the end of the reaction. So NAD removes 2 hydrogen atoms. Okay, so again, here's the hydrogen atoms. We're talking about 2 electrons in this case, 1 from each. From the original glucose molecule, NAD plus 2H, which creates NADH plus H. Okay, one of the hydrogens bonds to the NAD molecule. There's still a free hydrogen atom in the end, or a free electron, I guess you can say. And if you look down below, you could see the NAD plus form, which is the oxidized form of NAD. And then we have NADH, which is the reduced form. It has gained that extra electron from the hydrogen, and it's called NADH. When it has lost an electron, right, losing it makes it a little more positive. We call it NAD. So again, this is a prime example of how we will be seeing redox reactions occurring in these cellular processes when we create ATP or create glucose. There's also another molecule that is very tightly knit, associates the same way as NAD plus and NADH, and this is FAD. This is another type of electron acceptor. Okay, it can accept 2 hydrogen atoms, or 2 electrons in this case, so it can be either oxidized as FAD when it's lost some electrons, or it can be reduced to its form of SADH2, where it's gained electrons. Okay, we'll see this more in the Krebs cycle, but it's the same idea as NAD. So let's talk about stage 1 of cellular respiration, which is glycolysis. We've seen this word a lot. We have seen it on those diagrams already, and this is what we'll be talking about today. So we know that glycolysis happens in the cytoplasm, okay, outside of the mitochondria. And the word itself, glycolysis, glyco means sugar, and lysis meaning breaking apart or splitting. So this whole first stage of cellular respiration has to do with taking that 6-carbon glucose ring structure that we get from either consuming foods or that plants can directly make through photosynthesis. Takes that 6-carbon glucose structure, and through the 10-step process that glycolysis is, at the very end, we end up with 2 pyruvate molecules. Now, of course, scientists need to make everything complicated. Pyruvate is essentially half of the glucose molecule. Glucose has 6 carbons. Pyruvate is half of it. It has 3 carbons. Okay, so I guess you could think of it as a pirate stealing half of a glucose molecule. Okay, so it's called pyruvate. Now, although glycolysis simply starts with 1 glucose molecule and ends up splitting it in half into 2 pyruvate molecules of 3 carbon, there's a lot that happens to be able to do this. So throughout the 10-step process, a lot of people like to break it up into part 1 glycolysis or glycolysis 1. And this is the investment stage. This is where a lot of things behind the scenes happens to prepare the glucose molecule to split. And then glycolysis
part 2 or glycolysis 2 is the payoff stage. This is where we actually end up producing ATP. So let's look at the first 5 steps that happens in glycolysis. Okay, so we start with our glucose molecule. You can see on the diagram on the very top right, it's showing that 6-carbon ring structure of glucose. And it actually requires some ATP to phosphorylate glucose into a compound called G6P. Okay, this is actually a type of activation where the phosphate, there's 3 phosphates, right, on that adenine nucleotide. There's 3 of them, and 1 phosphate from ATP is added to glucose to form a new compound. Now it has a P on it called glucose 6-phosphate. That's what G6P stands for. This is an example of substrate level phosphorylation. Okay, a phosphate breaks off and joins with glucose. Step 2. So we have that G6P, that glucose molecule with a phosphate group attached because of phosphorylation. And it's rearranged, reconfigured into a form called F6P. Okay, so you can see on the very right-hand side of stage 2 there, you can see that glucose molecule. It's rearranged a bit to make it into a fructose instead of glucose. Remember, we can sometimes some functional groups can just flip from top to bottom, and it makes it into an isomer. So this is an example of isomerization, which kind of converts it from the G6P to an F6P. It still has that phosphate group attached, just rearranged a little bit in the compound itself. Then we have stage 3, and we see another ATP molecule coming in and phosphorylating that F6P. Okay, so it's taking that fructose 6-phosphate, still has the phosphate group, it's adding another phosphate group to it through phosphorylation. Okay, you can see that ADP ends up because it's taken a phosphate from it, and it forms fructose 1,6-diphosphate. The 1 and 6 are saying or telling us where the phosphate groups are attached. Now there's 2, right? Because it's just added another one. So that's another form of activation. Again, it's taking another phosphate from the ATP, and this is another example of substrate level phosphorylation. And then stage 4 and 5 kind of are shown together. This is another example of isomerization. Okay, so the fructose 1,6-biphosphate, which shorthand they call F16BP, it splits. And when it splits, it becomes dihydroxyacetone phosphate, or in shorthand, DHAP. Okay, so it splits into DHAP on one side, and the other is split into G3P. And this results in the end 2 G3P molecules. Okay, it's a little confusing because it's showing 2 things happening at once, but in the end of stage 5, there are 2 G3P molecules that exist.
So now we're on to glycolysis part 2, so steps 6 to 10, and this is the payoff stage. With in glycolysis part 1, that's the investment stage. We saw ATP coming in and phosphorylating a substrate and leaving as ADP, right? So it's investing some energy to then be able to make more energy in the second half, which is the payoff part. We're going to see ATP being produced. So in this part 2, we start off step 6 with G3P compounds. That was the product of step 5. Okay, remember, there's 2 of them that were produced due to splitting in part 5. So now we're going to be seeing 2 pathways, each of them being exactly the same. It's just to show us that and remind us that this is happening to each G3P compound. Okay, so we have those GP3 molecules, and they're converted into BPG molecules. And there's 2 of them for each G3P molecule that it starts with. So what happens is there's a redox reaction where each molecule of G3P is oxidized by that NAD+ coming in, and we see movement of electrons in that way. We also see that there is an activation portion that's happening as well. We see that little PI coming in at stage 6 in the diagram. That PI is telling us it's a phosphate group. So a phosphate group is binding to G3P, and it's now going to create a biphosphoglyceride, or BPG. That's where the bi comes from. Now there's an extra phosphate, and it makes it step 7 with the BPG. The BPG is now converted to 3PG. And remember, again, there's 2 of them on each part of the flowchart there. So what's happening is high energy phosphate groups on that BPG compound that starts at step 7. Well, there's 2 of them, and each will phosphorylate. They'll break off and join with the ADP from each BPG. And they're going to phosphorylate an ADP each to now make ATP. Remember, 1 on each side. That's why there's 2. Okay, so it's adding that phosphate group. It's breaking off from the BPG. It's joining the ADP. And now, bam, this is the first production of ATP in cellular respiration. Okay, stage 7 of glycolysis. So that's phosphorylation there. And then step number 8, we have our 3PG. Remember, there's 2 of them. And there's going to be an isomerization that happens. So the movement of functional groups to rearrange their order. They still have the same chemical composition, and through the isomerization, now they're rearranged to form 2PG. Now, those 2PG molecules are converted to 2PEP molecules. And this happens by the removal of 2 water molecules, a water molecule from each 2PG. Once it's released, it creates a new compound called PEP, or phosphoenol pyruvate. Okay, see the pyruvate there, which means we're almost at the end product. Remember, the end of glycolysis, it splits the glucose into 2 pyruvate molecules, and we're almost there. Okay, so when there's a removal of water in this stage, in stage 9, this is called dehydration, which changes it to PEP. And lastly, for glycolysis, PEP, and there's 2 of them for each of those flow charts there, are converted into a lump sum of 2 pyruvates. Each PEP produces a pyruvate. The high energy phosphate groups on the PEP, just like up there in step 7, there's high energy phosphate groups attached, and they detach from the compound, in this case from the PEP, because there's ADP that interacts with it. It takes some of the phosphate group from each one, and bam, there's another ATP. Again, this is happening for each compound, so there's 2 more produced here. Okay, so again, this is phosphorylation, and in the end, we end up with pyruvates, 2 pyruvates. But more importantly, from this second part of glycolysis, 2 ATP. Okay, so I said that the second part of glycolysis, in the end, we end up with 2 ATP. And that's because although we produce 4, the first half uses 2. Okay, so remember, glycolysis 1 is the investment stage. It uses up 2 ATP to be able to transform that glucose to eventually 2 G3Ps. And then in stage 2, those 2 G3P molecules can generate through phosphorylation and isomerization and dehydration, eventually 4 ATP. So again, glycolysis 1 uses 2 ATP, glycolysis 2 produces 2 ATP from each G3P molecule, so it ends up being a lump sum of 4. But if we subtract the investment piece from glycolysis 1, 4 minus 2 will give us 2 ATP. We also, although we keep focusing on ATP, we also have to remember that NADH is a high energy molecule as well. And through this process, we saw redox reactions happening where NAD plus transformed into NADH through the movement of electrons. So let's not forget about the 2 NADHs that are created as well. The overall reaction of glycolysis can be expressed in the chemical equation shown here. If you notice, there is no oxygen seen throughout this process, meaning it is an anaerobic process. Help yourself remember what is happening in redox reactions with this following saying. Leo the lion says GER.